All right. So welcome everyone to the second part of this webinar. I will be talking about overcoming scattering effects and absorbance effects in micro-FTIR imaging data. My name is Benedikt Hufnagl. I'm founder and CEO of Hufnagl Chemometrics, which is an Austrian software company that specializes in the field of chemometrics. And our main product currently is microparticlesAI, which I will be talking about a little bit, and also, of course, the mentioned effects that we encounter in FTIR data. Um, yeah, so in today's webinar, we talked a lot about, and still will be talking about, artificial intelligence, microparticles AI, and the topic that I'm talking about is located in the machine learning domain, which is a subfield of AI. And deep learning is also part of this domain, which has popularized this area of research. And in microplastics analysis, we have two key tasks, you might say. One is the chemical characterization. So if we're dealing with spectra and we want to identify plastics based on the spectra. And the other thing that we need to deal with is particle identification. This is often done using microscopy data. But in today's presentation, we will do the particle identification based on the chemical spectra, as you will see later. microparticlesAI is a software tool for microplastics analysis workflows. So what you do is in your lab, you start with the devices that you have, some imaging capable device like this. You load the data into the software. It applies machine learning to your data, recognizes the particles, and then summarizes and aggregates the results as a report. And using this app, you can do the whole analysis workflow. So we provide various interfaces to different device vendors. So there's typical imaging devices, but also for the ones that create much larger files like the Agilent Carrier 620 and the DRS Spero QT. So you can analyze very large data sets, 70 gigabytes or more. We provide different kinds of machine learning models for environmental applications, food and beverage, textiles, and many other things. And all these are aligned with the current regulations in the EU and the ISO standards. There are tools for auditing particle data, ensuring quality. So you also have the option to check every particle, compare it to reference spectra, and so on, as mandated by these two ISOs here. And of course, you can do reporting. So that in a nutshell is the software microparticlesAI. And now let's dive in on some problems that we often encounter if we apply this kind of software to micro-FTIR imaging data. So I first will be talking or do a short recap about machine learning, really super short. Then I would like to show you some examples about total absorbance, some issues with fibers, and scattering. And the motivation behind this presentation is that when I interviewed users of the software, I realized that many people struggle with these kinds of effects. So they are not aware of them, that they are there, or they don't really understand what am I seeing in this data here. So this is why I would like to show some examples that you might recognize them as well. First, let's start with machine learning. So in a nutshell, what we want to do is we want to train a function to do a certain job for us. In the case of microplastics analysis, we wanted to recognize spectra. So this spectrum shall be recognized as polypropylene, this one as polystyrene, this one as PET. And within this black box, this magic happens. What do we have to do in order to manage this? First, we need a lot of training data, so many annotated spectra, and then we let machine learning do its magic. It deduces a decision boundary in this high-dimensional feature space, like Friedhoff already told you, and then this model is ready for deployment within a software. And if we want to apply this to imaging, what we can do is we get different kinds of data from our machine learning model. First, the highest matching polymer class, but also some kind of scores for every spectrum. And if we apply this machine learning model to every pixel of our imaging data set, we can get a classification result like this, where the different particles are highlighted in the colors of the respective polymer class. We can also look at the scores that the machine learning model provides. So here you see lighting up the polypropylene particles, the PE particles, the PET particles, the PS particles, and so on. And if we're lucky, we also have a microscope image that we can use additionally. So now let's have a look at the main topic of this presentation, the physical effects that can be problematic when analyzing micro-FTIR data, total absorbance. So total absorbance happens if we push our devices to the upper size limit of particles. And what happens here is that the illuminating radiation that passes through the particle in transmission mode gets absorbed by the particle. So here is an artificial spectrum that you can see here. And if this spectrum suffers from total absorbance, what happens is that the peaks get cut off here and here, for example. But we will also see that these peaks, they get, of course, larger, the smaller ones. And here you can see a real world example from a very thick particle, which is rather thin at the edge, but the thickness increases towards the center. And now I will sample across this particle some spectra. So here you have a very nice polypropylene spectrum. And already the next spectrum, you can already see the total absorbance effect. So here the peak is cut off. These are still complete. And here the situation gets worse. But on the other hand, we get new peaks which can be used for classification. Here almost all data is lost. Here still something is left that we can use for classification, and here we only see noise. Now the question is, can machine learning still deal with this kind of data? Yes, if you train the models well. So here you can see a result from a convolutional neural network, and you can see up to this spectrum. So this is the one in blue. The model is still able to recognize this as polypropylene. Then it turns gray, the color, because this reflects the class other, which is unknown. And if we compare this with other kinds of machine learning models, even though they are trained with the same kind of data, they behave differently. So for example, here we have random forest, which was also mentioned before. And you can see the result is slightly different. So the capabilities between models vary concerning these effects. Now let's have a look at the real-world application case. So this is a large environmental sample measured with the Bruker Lumos 2. And you have some very thick particles here. So here this is a zoomed-in view of that part. And now you can see the result from the CNN. You can see that these particles are still kind of recognized well. And now let's compare that with the random forest. And you can see that here this is just a noisy cloud. It's still able to recognize it, but it's not as competitive as the convolutional neural network. All right. Next thing is fibers and the focal plane. So fibers are also very common in the environment, but also, of course, in textile samples. And the problem with fibers is that they are not flat on the filter, and therefore they are not aligned with the focal plane. And this, of course, is a problem because you get this out of focus spectra. And how that looks like, we're going to see in the next slides. So here you have a wastewater treatment plant sample from a textile plant. And on the right-hand side, you have a textile washing sample. And what we want to do is we want to recognize all the fibers in these samples. And of course, many of them will not be within the focal plane. And here we will take a closer look at how this looks like. So we will look at this fiber here. You can see in the microscope image that on top, we have a nice focus on the fiber, and here it gets really diffuse. And again, we will sample across the fiber. So this is a nice polyethylene spectrum. Also, the next spectrum, I would say, is still kind of okay. You can recognize it well. But as further we go, the signal decreases more and more. The peaks get weaker and weaker up until until this point where there's almost no signal left. And now the question is, can we still detect that with machine learning? Yes, so in this case, it is still possible. So machine learning models have very powerful capabilities concerning these effects and also dealing with very low signal to noise ratios. Now to finish up this presentation, I would like to take a look at µScattering. This effect occurs if we have very small particles or fibers, and this effect occurs if the size of the particle gets into the size range of the illuminating wave numbers. And then the photons are scattered by the particle. And these causes two kinds of effects. The first one is what I would call the classic µScattering effect. So if you look at this example spectrum here on top, if it's affected by µScattering in the classical sense, you get these strong distortions of the baseline. So it moves around like this. Actually, that's often not that problematic for machine learning. It's rather easy to deal with, and still you will be able to recognize the spectra well. A much more problematic thing is the resonant µScattering effect. So here I'm using a straight baseline that it's easier to recognize. But what happens for peaks on the right side of the spectrum is you get this shift. So the peaks seem to move towards the right-hand side, and you get this strange S-curve. If you want to find out more about the theory and background of this effect, please have a look at this publication. It describes it very well. And now let's have a look at the practical example again. Here you can see some kind of polyester fiber from an environmental sample. You see these typical prominent peaks of polyester. And now let's see what happens if we again sample across this spectrum. So here we already can see a strange change in the spectral signature. And now it gets even worse. So the peak gets this very prominent S-shaped curve. And then it actually inverts. So the peak is completely lost. It disappears and turns into the other side. And yeah, here it's even worse. So now you can see all these spectra superimposed. But if we train our machine learning models well, we are still able to detect these fibers completely. All right. Then I would like to mention upcoming workshops. If you're interested in trying out the software, there are two opportunities in July. One will be in Australia pretty soon, so just 2 days left to submit your abstract. And the other one will be in Singapore. So I'm looking forward to meeting you there, and thank you very much for your attention.